Okay, morning everyone. So we're going to go ahead and start the webinar now. We have some more people joining, and we'll let them in as we go. But thank you all for joining us today for this fourth episode of the EcoGen webinar series. Today we're focusing on advancing animal health through microbiome research. And we'll be joined today by Amalia Bogri, David Yáñez, and Bryan Wang. And this is again the fourth episode, and this EcoGen cluster is a research cluster led by HoloRuminant, and is joined with livestock, Geronimo, Rumigen, and 3D-omics. So first we'll have a bit of introductions. In the next slide, we'll go over some house rules. Then we'll have a presentation from David. One thing to note here is that David will need to leave latest at noon. So in an hour. So if you have any questions for his presentation, do drop it in the chat during his presentation or before he leaves, so he has a chance to answer your questions before he needs to go. And he is today joining us from the HoloRuminant project. He is also involved in the livestock project, but his presentation today is research from the HoloRuminant project. Then we'll be joined by 3D-omics colleagues Amalia Bogri and Bryan Wang. And then afterwards, we'll have an open discussion floor where you can speak openly and discuss with each other. So some house rules. If you're not speaking, do please mute yourself. You will have the opportunity to speak, but if you're coming in and you're unmuted, do please mute yourself. If you have any questions or comments, we do really want you to put them in the chat, so we can have an open discussion and answer any questions you might have. You can also raise your hand, and then after a presentation or after we're done answering another question, we'll call on you when it's your turn to speak. We are also recording this session. It will go on the HoloRuminant YouTube channel afterwards. So if you don't want to be recorded, simply just turn your camera off. And if you have any technical issues throughout the webinar, please, there we go. And then if you have any technical issues, you can pop a question in the chat, or you can also email us at projects@efab.info. That will work as well. As I mentioned a bit earlier, the EcoGen cluster is led by HoloRuminant, and this is the final episode before our summer break in the webinar series. So if you want to be notified for when we start up again, do register for the HoloRuminant newsletter. We'll also have a link for that in the chat. And as mentioned earlier as well, this cluster is a combination of several projects. So if you want to learn more about the individual projects, you can scan this QR code, or alternatively also look at the link that we're sharing in the chat. And that's it from me. I will go ahead and hand over to David, who will do his presentation.
Hello, everyone. Good morning. Can you see my screen?
Yes.
Good. Well, thank you for the presentation and thank you for the invitation. It should not be me who is presenting this work. This is part of a PhD of Eva Romero Recio, PhD student here at CSIC, but now she's traveling, and we are involved in livestock, but this is a work that we're doing within HoloRuminant as part of Work Package 3, which is a work package that addresses different health and animal welfare issues in ruminant production systems. And there's a particular task working on young animals and the management of the different systems and the impact that modulating the microbiome may have on the overall animal health. So this is part of that task. And in particular, we're going to be talking today about using feed additives in dairy calves that are used for beef production, which you will see in a minute is a very relevant in the livestock production system in Spain. So before we go, let's see if I can navigate through before we go to the actual experiment that we conducted in HoloRuminant and some of the results, I'd like to take you through some important issues in relation to the management of young ruminants. So we're gonna just, for those that are not that familiar with the rumen microbial ecosystem, we're gonna just go very quickly through a presentation of the importance of the rumen microbiome, and then in particular the processes of the colonization and development in early life in ruminants. that will then take us to some of the challenges associated to the use of dairy calves in beef systems. And then eventually we will present some results of our experiments in HoloRuminant. So if we start with the ruminant microbiome, I'm sure you know that ruminants are herbivores, and they establish a very interesting relationship with a complex and diverse microbial community comprised by bacteria, archaea, protozoa, fungi, and also phage, which are the ones that ferment and degrade most of the nutrients that come into the rumen in the diet, in the forage, the grass, or the concentrate. So carbohydrates are further degraded into sugars, and then microbes obtain energy through that degradation, and that generates volatile fatty acids, which is the main source of energy for the ruminant. And something similar occurs with protein, and then the amino acids and also the ammonia is used by the microbes to produce their own proteins. And in fact, this is the main protein source of the ruminant host. So the byproducts of the fermentation, BFA and proteins from the cells, for the microbial cells, are the main sources of nutrients for the ruminant. If we could open a little hole here on the left-hand side of the ruminant and take a look on a microscope, we could see something similar to this. Not exactly the same, because this is the microscope only can let us see part of the diversity. These are protozoa colonizing feed particles that have been ruminated by the animal, and then they are degrading these components of the feed and then producing some of the metabolites that we were talking about before. And I like using this video with not only with farmers but also nutritionists, so to tell them that when they think they're feeding an animal, a cow, they're not actually feeding a cow, they're feeding microbes, and those microbes are the ones that feed the animal. And this is something very, very important to keep in mind. This microbial ecosystem is, of course, very diverse and complex, and has two main characteristics that make it very peculiar. One, that is very redundant, and that means that there are different species and microbial groups that can undertake the same functions. So they are prepared for a diverse type of different scenarios in terms of feeding systems and problems they may have in different seasons of the year. And the microbial ecosystem is also very resilient. So it's very difficult to modify the compositional activity in the long term. And when you stop any manipulation, normally it goes back very, very quickly to the original situation. So it's a very interesting, but at the same time very challenging microbial ecosystem. And it's challenging because and difficult to modify, because it's been evolving together with ruminants for such a long time. So if we take a look at the evolution of and when the first ruminants as we know them today from the family Bovidae were present in our planet, they were like 10, 15, 20 million of years is when they were present first in our planet, because the forage or the grass production, the climatic conditions allow these animals to be in the planet. And since then, the microbial ecosystem has been co-evolving with the animal for millions of years. And if we think about when did we start to try to modify not only the animals but also the microbiome through first domestication, but more specifically through intensification, the time that we've been dealing with that microecosystem is very short comparing to the time that it has been evolving with the ruminants. And I explained some of the challenges that we face when we try to modulate the activity of this microbial ecosystem. And one of the aspects that are very relevant is when and how these microbes get into the rumen. And this is just a graph illustrating how the rumen develops. When the ruminant is born, it's not a ruminant. So the rumen only takes around 20-25% of the whole gastric system. And then it takes a few weeks until the rumen develops to the full size, which is around 80% of that of the whole gastric system. And there are different phases going through that process, like first no rumination. So the first 3 weeks the rumen is not active. Then there is a phase in between of transition, 3 to 8 weeks, when the rumen is being developed and some microbial activity. And then after week 8, rumination starts to become active, not fully developed, but active, and it's normally when animals are weaned. And in those 3 phases, there are 3 parallel processes occurring in terms of the rumen development: the anatomical, the functional, and the microbial. They go hand in hand together, and equally important. And now in the HoloRuminant, we basically focus on the microbial part of this development, which is very important. When the animal is born, it's not sterile, but the rumen that is not yet a rumen, it starts to be colonized from birth. There are also studies that suggest that maybe there is some colonization pre-birth in utero, but what we know is that the animal gets colonized when it goes through the vagina, and then from the first time it's out of the mother, then it starts being colonized, and in 1 month or so, the microbial biomass in the rumen is similar in terms of concentration to the adult animal. And here there are many studies that have been trying to describe this process and the factors that influence this process. And one of the main factors is the contact with other adult animals. And here we can make a very clear distinction between systems where the newborn stays with the mother and is raised on natural milk feeding, and those that are taken from the mother after birth and then kept in isolation with no direct contact with adult animals, and they're fed meal replacer. This distinction is very clear, and we know that most dairy systems operate under this artificial system. So the animals are giving colostrum, but then they're taken away from the mother, and they're normally kept first in isolation and then in small groups with other small animals. While in natural systems, mainly in beef, some of the beef systems, the young animal stays with the mother for 2, 3, or 4 months. We've been working on and trying to describe and compare these 2 models, and we have clearly seen that when we compare natural and artificial meal feeding systems during the first month of life, this is a graph showing the colonization of different species of the rumen in natural and artificial meal feeding system. This was done in dairy goats, and we could see that clearly the animals that stays with the mother and have direct contact with them, they have a much greater biomass of colonization as they grow, and that is reflected into a quicker and greater microbial fermentation. So if we go from day 1 to day 28, we can see that the concentration of volatile fatty acids goes up much quicker in animals that are fed that stay with the mother. So and there are many other studies, and there's also studies, of course, in humans as well. They show this, and this is important because, as I said before, in modern dairy systems, animals are taken from the mother. So the management system really promotes a deficit in terms of microbial colonization that may have some natural implications to the development of the rumen, and also eventually to the health and the well-being of the animal, which is what we are discussing today. And there is another issue which takes us more into our actual work, which is the fact that the male dairy calves, most of the male dairy calves are taken for beef production, and this is a part of the beef sector in Spain is quite important. So in Spain, there are lots of male dairy calves that come into the country, or they stay in the country from dairy farms, but also they come from other countries. And these calves are taking like 2-3 weeks of age, so they still lactate in milk feeding. Then they're taken to assembly centers where they are taken grouped together with animals from other farms, and then they're classified according to morphology, weight, etc. And then they are transported, in our case, from France or other countries into the growing and then the fattening premises. And this is important in terms of the age of the animal. So the animal is still milk feeding, so that cannot eat solid feed. during this process. This process takes 1 week max, and in Spain there are like 700,000 calves coming every year, and 65% of them come from France. So this is a big part of the sector, and the whole management process has implications in terms of probably not optimal growth, because the animals are subjected to some stress, transportation, being grouped with other animals, reallocated to new environments, and then as a consequence for stress and not very good or potentially not good feeding, digestive disorders, and respiratory infections. So that's the context where we're working. So in that context, we perform a work in HoloRuminant first, trying to see whether the transportation from France into Spain had an impact on the health of the animal, and then assess one of the strategies that we could use to compensate that deficit of microbial colonization and rumen development that we've been seeing before, which is the use of feed additives. And this is a work that has been recently published in Animal Feed Science Technology. So if we go to the effect of transportation, very, very quickly, what we did was go to the assembly center, so where all the young dairy calves of 2 or 3 weeks of age are taken and grouped together. They stay there for 2 or 3 days. They are classified into different groups. Then they're going into the lorries, and then they are transported to Spain. In our case, it was a 750 kilometers and 12-hour journey. That was in 2022, I believe, and it was in October, which is important because October is relatively a good time of the year for this transportation. It's not cold, it's not hot. So we took blood samples and from these animals on before departure, and then upon arrival to the farm. And I'm not going to show all the results, but just as an example, we could see that there was no clear sign of stress in terms of cortisol concentration. There was slightly numerically higher in animals. This is before and after, before and after, and before and after. In cortisol, there was a slight increase. It was not significant, but considering that these animals have to be transported and then downloaded into the premises and then taking the blood samples, then that may explain that slight increase in cortisol. The main feature that we could identify was a sharp decrease in glucose in blood, and that is basically a reflection of animal fasting and not eating during the journey. And in terms of because it was not very hot time of the year, we couldn't see any signs of dehydration, iron, creatinine,
allora è una Porsche che al momento
and many other parameters. So the main conclusion of this part of the study was that animals were in good condition when they arrived to the farm, and the only problem they may have was obviously the fact that they were very hungry because they did not eat during the journey. So that's the transportation. And then if we go into the actual trial where we tested the different feed additives, where we used 112 male suckling calves. They arrived 3 weeks of age to the growing farm in Lleida in North Spain, and then we split them in 4 groups: a control, and then 3 treatment groups that received different feed additives. And the feed additives were provided to the animals from the arrival to the farm to day 45 when they were weaned. And then from day 45, they all received only one of the treatments. And I will explain the treatments in a minute. So we had 28 animals in each experimental group. And pre-weaning and post-weaning on day 35 and 105, we took blood samples and rumen samples to analyze to see the evolution of some of the more important physiology parameters, and also rumen fermentation and rumen microbial ecosystem. So the treatments that we applied were either controls or no treatment. So there were animals that for the first 45 days did not receive any additional microbial or any treatment at all apart from the diet. And then the other 3 treatments were EO were a blend of essential oils specifically designed for this type of animals. Then another group that had 2 strains of yeast probiotics, and I will go to that in a minute. And this is the SIM group. And then the mix, which is a mix of lots of things, like a kind of cocktail. And this was the actual mix that the farm, the cooperative were already using. And that included essential oils, yeast probiotics, and also butyrate. And then again, as I said, and this is important, after weaning, all animals were changed to the mixed diet. Then just one note on the probiotics, which were included in 2 of the treatments. You know that yeasts can be used as probiotics. In this case, they are Saccharomyces cerevisiae. They are not symbiotic animals in the microbes. In the rumen, so they have to be provided to the rumen daily. But their activity is very interesting for the rumen, especially in these situations, because they consume oxygen, which is something that we want to avoid into the rumen, so they keep anaerobic conditions. And they also promote the growth of bacteria that can use lactate, so that helps to maintain pH in the rumen and also produce butyrate, which is very important for the development of the epithelium in the rumen and in general in the digestive tract. So that's the reason why we use the yeast probiotics. Then the blend of essential oils is one of the commercial practices that it is quite generalized at the moment, so we opted to use that one. And then butyrate is another product that is also being used basically because, as I said, it helps. It's a nutrient for the epithelial cells, so it helps the development of the epithelial part of the rumen and therefore the absorption of capacity of nutrients. So as I said, we took rumen samples and blood samples pre and post weaning. So if we take a look at the pre-weaning results, we could see that the total BFA, the volatile fatty acids concentration in the rumen was not significantly different in those animals, but we could see an effect on the concentration of beta-hydroxybutyrate in blood. Beta-hydroxybutyrate comes from the absorption of butyrate and acetate to some extent in the rumen. So that was an indication that in the rumen there could be a greater activity pre-weaning of the animals that were treated with the 3 different additives. If we look at the biomass of the concentration of microbial groups in the rumen, we could see that there was no difference between treatments in Bacteria and Archaea. They were fully colonized, normal figures for animals, quite high actually. So there was a good colonization. But one important feature is that there were no protozoa, neither fungi in the rumen of those animals. So pre-weaning, no protozoa and no fungi. If we move now to the post-weaning, which is 2 months after weaning, and when all animals were at this stage were under this diet, but pre-weaning they received different treatments. What we could see is that there was a significant difference in BFA concentration in the rumen, so greater values for the animals that received the treatments. Again, beta-hydroxybutyrate was in some cases significantly higher compared to the control animals, so indicating a greater microbial activity in those animals compared to control, even though at this stage all animals were under the same diet. And then in regards to the microbial population for the abundance of the different groups, again, no differences in Bacteria and Archaea. In fact, the figures are larger than pre-weaning, which is normal. Fungi were present. They were fully colonized by fungi, which is very important in terms of fiber degradation. But the absence of protozoa persisted. So these animals did not have any protozoa in the rumen. And this has been obviously described before. So animals that are not in contact with adult animals during the early life, and if they are not in contact with other adult animals later on, they stay free of protozoa. And that has important implications in terms of the microbial fermentation. And there are some adaptations to compensate the lack of these microbes, because they can account for nearly 50 or 60% of the total microbial biomass in a normal standard rumen. Then you could ask, well, what about the performance of the animals? Well, we could see no effect on weights or average daily gain, and this is reflected here. So they were weighted different times, and when we calculated the average daily gain for the first period, around 700 in daily, 1,000 grams or 1 kilo or nearly 1 kilo towards the end. There were no differences. The only difference that we could observe was and this was pre-weaning and post-weaning, the standard deviation was always greater in the control animals. So showing that the animals that were under the treatments were all, as groups, were more homogeneous, so that they were all more close in weight. So they were not like in control animals, some animals that were larger and some animals that were smaller. So there was a more homogeneous evolution of the animals in terms of body weight gain, which in a large farm can be very beneficial in terms of managing the groups and then sending them to the slaughterhouse, etc. And then this is something that we are analyzing at the moment, just the composition of the microbial ecosystem. So this is just preliminary results. So this is on bacteria. Samples were sequenced, amplicon sequencing, and we could see in terms of the alpha diversity, no differences between the groups pre-weaning, but some slight differences post-weaning. Not great, but some differences. We could see that there was a strong core microbiome right from pre-weaning that was common to all microbial groups. So only a few microbes were only present in some of the treatments, so showing that right from the beginning, the pattern of the colonization was very similar. And if we look at the PCO of the break cartilage distances pre and post weaning, we could see there was no clear separation pattern. So all populations were apparently very, very similar. When we did the discriminant analysis pre and post weaning, then we could see some separation. So we could see that the bacterial population was different in animals that received the mix and the symbiont treatments compared to control and essential oils. And then post-weaning, there were differences, but in this time, symbiont and control were different. So something that we're still working on to try to explain. But what we could see is that some of the differences that were here persisted here. And the fact that 2 months after having the same diet, there are still differences in the microbial population between the two groups show that there is some persistent effect of treatments if they are applied early enough in diet, regardless of the diet or the management that the animal receives after weaning. What we're doing at the moment is trying to identify what microbes are actually different between some of the groups pre and post weaning in terms of relative abundance, and we have identified some that are related to butyrate production, but this is work that we are now undertaking, and we don't have final results yet. But I think we need to dig deep into the different specific microbial groups that can show why the fermentation profile was slightly different in those animals pre and post weaning. So to conclude a few things from the work we've done, it's important to, now that today we're talking about animal health and well-being, to address the risk associated to the management of dairy calves for beef production, and this is important because also there are societal concerns about these systems in which animals are transported very early and then taken away from the original farm, etc. In our situation, probably because the management was correct and optimal, and the time of the year was also not challenging in terms of temperature, there was no major effect of transportation, apart from the animals fasting for a long period and obviously a deficit in glucose, but they were recovering quickly. In our trial, we could see that feed additives could promote rumen development in early life. So there was a greater fermentation activity in the rumen of those animals that unfortunately did not translate into better production performance, slightly better in terms of homogeneity of the group and not the growth. And what is important, though, is that whatever treatment we apply to help to promote a better microbial development in terms of the microbial population of the rumen, they need to be applied pre-weaning when the process of colonization is occurring. If we do that after weaning, as you could see, the control group had no treatment before weaning, but they received the additives after weaning, and they still were different to the other groups. So the treatment has to be applied in that short period of time, that window of time, where there is more plasticity in the rumen microbial ecosystem. And with that said, that's all from me. I'd like to thank, obviously, Eva, who did most of the work, and Eva Ramos as well, co-supervisor of Eva, and the whole group that worked hard to get all the samples analyzed and the results I could show you today. So thank you.
Thank you, David. So we've got a couple of questions coming in. So if any of you want to say them out loud, do feel free to put your hand up. But we've got one from Aurel here. That is, could you please re-explain the reason of protozoa, if I'm saying that right, absence at a 2-month post-weaning? And could you please supervise HDR in France?
Right. Well, the first question, yeah, the reason why protozoa did not colonize is because the only way that those microbes that are very sensitive to being exposed to oxygen, and they cannot develop any system to be floating alive in the water or on the ground. The only way they can be inoculated into the rumen is by direct contact between an adult animal and a young animal. So it has to be mouth-to-mouth contact. So if those animals are taken away from the mothers in the first hours where the rumen is not developed, and then they don't have any contact with other adult animals, then that results in no protozoa at all. And that has important implication. It can have positive and negative. And the other question, could you please supervise HDR in France? I don't know what HDRR is, but I'm very open to discuss.
Yeah, Aurel, if you're here, you could speak up or put it in the chat.
Yeah, or send me an email.
Oh, can I have
put
your
my email
email?
address in the chat.
Yeah. HDR is habilité à diriger la recherche.
Okay.
I think it's a typical French degree or
okay.
habilitation for supervised research after PhD or postdoc.
Okay. Yeah, I'd be very happy to see whether it's possible. My French is not very good, but maybe I'm sure we can understand each other.
Yeah. Okay. So I will email you.
Okay. Yeah, I put my mail address in the chat.
Yeah, and also regarding the reason you just explained for protozoa, my point was 2 months after weaning, the animals will remain without any protozoa. I'm quite surprised about that because I've seen some animals after sampling away from the parents with some protozoa after 2 months. So they were in anaerobic conditions and everything was going smoothly in terms of microbial, the overall microbial composition and structure regarding protozoa, bacteria, archaea, fungi, and so on. Yeah.
Yeah, without going into detail, it's normal that there are no protozoa if they don't have contact with adult animals or contact with farmer that has been in direct contact with adult animals previously in minutes of difference. Because as I said, the only way this and there are lots of flocks in the world that are free of protozoa, and that's the reason why. If they get in contact with adult animals, they get colonize in very, very quickly.
Yeah.
So yeah, it's normal that they become free of protozoa, and that changes the whole microbial ecosystem for many reasons.
Yeah, and did you also have you seen some impact or effect on the digestibility or even
No,
methane
we didn't measure
production?
digestibility. We just took rumen samples and analyzed the rumen fermentation. Just one note to clarify, I can see a message saying that 2 parameters, the average daily gain was lower. No, these figures are about the standard deviation, not the average daily gain. So the average daily gain was the same, between 700 and 1 kilo per animal a day. And what was different was the deviation of those values, and it was created in control, not in the animals that were treated. Just to clarify to the comment from Angela.
Okay. Yeah. Thank you.
Thank you.
Okay, great. Thank you, David. Then we have a question from Jessica. You've got quite a detailed question. Do you want to maybe speak up and say it?
Hi, David. Thank you for the
Hello.
talk. It was very interesting. As we're also working in the rural livestock with the calves and the rumen sampling, I was wondering, how was the rumen sampling done? Was it also like with the tubing in the esophagus, or how did you do that?
Yeah. I didn't have time to discuss that in detail. But animals were sampled using the stomach tubing, and that was done in 2 days. And the groups were split equally, so there was no influence of the 2 days into the experimental groups. And that was done. I think it was before the morning feeding, because if you wait to have 2 or 3 hours after feeding, and you have a large number of animals, then you're always going to have a variation across animals, because some may be like 2 hours after feeding, and then another one 3 hours, and then you may have a variation. So sample was done early in the morning before feeding, and in 2 days. So because if you have to do 112 animals in one day, it takes hours, and that's too long.
Yeah. Okay.
And it was done just once pre-weaning, and then post-weaning 2 months after.
Okay. And after the weaning, they were fed TMR, or what kind of feed did they get?
Yeah. They fed in this farm, and that's also known in most of Spanish systems. They fed first. They had a very smooth starter concentrate, and then they have oat hay ad libitum, and then they changed the concentrate to another concentrate for post-weaning, and again hay ad libitum, and both were ad libitum, so there was no TMR.
Okay. Yeah. Great. Thank you.
Great. Then we had a question from Gina. Now, I'm not sure if you answered this already, David, because it was the comment you answered earlier was on this question. But it was, can you elaborate on why the feed additives didn't translate to improved weight gain despite showing improved rumen fermentation?
I cannot have a clear explanation. I think I don't think the changes in the microbial population were large enough to cause an increase in the growth. And I think that in this farm, this was a commercial farm, but in experimental premises that they have for these young animals, and we could see the management was quite good. So we could see that there was no much that we could improve, to be honest. So the slight improvement in the microbial population was not translated into improved in weight gain. Whether we could have a better result or better improvement if the animals were challenged in other periods, like becoming more stressed with very cold or very hot in the middle of the summer, maybe we could have seen a clear translation. But yeah, that's all we can say.
Okay, thank you. So then we have 2 more questions for we'll just try and get a few, and then we'll let you go. So Andres asked a question in the chat. Could you elaborate why the p-values of bacterial community were not significant for pre and post treatment?
Well, essentially because I think they were very similar, and there was only, and that's what we're trying to do now, trying to identify what was different in those, because in terms of biomass of the abundance, they were the same. in terms of the core microbiome, they were the same. And we can see now that some species are different, and these are the ones that we think that make the difference. So we're trying to identify them and then trying to correlate with the blood and the rumen fermentation parameters and see whether we can identify which ones are maybe making the difference or the slight difference in this case.
Okay, super, thank you. And then Angela, I see you have your camera on. Did you want to speak? Sorry for the call out.
Hi, David. Thank you for the
Hello.
presentation. I was wondering in general, how does this average daily gain differ in calves with and without protozoa? Because I have always in mind this textbook knowledge defaunation has no effect on digestion in ruminants, but
It depends on the
yeah.
diet. That's because obviously nature put protozoa there for a reason, so they are not there randomly. And as I said, they can represent half of the whole microbial biomass, so that they're very, they can be very important. If the diet is very high in fiber and different types of fiber, normally you have lower digestibility in animals with no protozoa because they have a big enzymatic arsenal to degrade fiber. Also, of course, fungi and bacteria do as well, but that's one of the main roles they do in the rumen, contribute to the degradation of fiber. So if the diet is high in forage or low digestibility fiber, forages, then it's better to have protozoa. And the other advantage of having protozoa is that when the diet is really high in soluble carbohydrates, or especially very high in soluble starch, they retain those starch particles and they slow down the degradation so that you don't go into acidosis. But none of those two situations was in our dietary conditions.
But in your case, now there were no protozoa, but the average daily gain is still comparable to other studies. I mean, it will not impact the body weight of these animals in the long term. No.
And as I said, there are lots of farms around the world that have ruminants with no protozoa, but we don't know. So farmers don't know.
Yeah. But in Spain, how much concentrate you feed to this? Is it 90 like feedlot?
It's kind of feedlot, but then it's not only the concentrate that you feed, but the composition of the concentrate. And if you could see the pH values, we were expecting much lower pH values in the rumen.
I forgot how was the pH?
Expecting values below 6, and we had take a quick look. Pre-weaning, it was 6.1, 5.9, 6.1, and 5.4, and then post-weaning, it was all above 6.5, so 6.8, 6.6, 6.4, 6.4. So we couldn't see any sign of too much being fermented and very quickly. And in fact, there was no incidence of diarrhea or animals that stopped eating. It was a very well managed farm as well, so which probably could play against us in terms of trying to to see a better or much greater benefit of feeding the feed additives. So my conclusion is that feed additives are not, in this situation, are not essential. So animals can live without feed additive, of course, but whether you use them to prevent some problems in some challenging situations is another question. So we're trying to see if we can repeat the trial in other conditions.
Thanks.
Okay, I think we managed to get through all the questions in the chat. Thank you so much for joining us today, David, and for your presentation. It was very interesting, and I think we had a very good discussion with the questions. So thank you.
Thank you.
Thank you.
And we'll hand over to Amalia and Bryan from the 3D-omics project, who will have a joint presentation.
Okay. Hi, good morning, everyone. I'm Bryan, and my colleague Amalia and I will be presenting a bit about this project that we're in called 3D-omics. And so kind of different from what David had shown, we are more of a methods development type of project. And so we focused a bit more kind of in a zoomed out way on the methods that we were developing. And so it wasn't too results heavy, but feel free to put the questions in the chat. Yeah, but the title of our presentation is How We Can Spatially Resolve the Multi-Omic Landscape of Animal Gut Microbiomes. And so in my postdoc and actually in my PhD, I did a lot of outreach, talking to more generic audiences, classrooms, etc. And I always like to start my talks with the concept of scale. And so I like to ask the classrooms and the students, what really is the smallest thing that you can see with your naked eye? And the general consensus is it's something like a human hair, or in the spring like now, where you see pollen coating your phones and your screens, etc. And so on my slide, I'm showing scale going from left to right, left being the biggest thing. And as you go down a scale, you go to the right. And so bacteria and viruses are basically things that you can't really observe unless you use a microscope. But in actuality, they're everywhere. And so another question I like to ask the students is to think about where exactly do you think there are most number of bacteria? And so oftentimes the students will give me answers about skin, about the mouth, about the nose and the ears, because that's what you think about. But in actuality, and most of you probably in the audience know that most of the bacteria that we find in our body is actually in our gut. And so it's estimated that somewhere up to 38 to 40 trillion bacterial cells, depending on before or after you go to the bathroom. And that is a lot of bacteria that are in our body. And so this is likened to the number of animal cells, so human cells that are in our body. But oftentimes we don't even think about it. When was the last time you thought about like, oh, how are my microbiome doing? How is my stomach doing in terms of bacteria? So in the project of 3D-omics, we don't study humans, we actually study livestock. So our focal species are chicken, turkeys, and swine. And so it is also known that maybe the numbers are not as high as that in the human body, but most of the bacteria that you find in these livestock are actually in their gut. And so this is also true for the rumens that David was talking about. And so in general, most of the times when we study microbiome, a lot of us are using poop samples as a proxy for what is happening in the gut. And so we endearingly call each other poop scientists, because what comes out of poop is majority what's undigested, but also the bacteria that comes through your digestive tract. But you can imagine when it comes out of the animal, it's often already, you no longer have spatial information. And also if you take gut samples from an animal, usually the first step you do is homogenization. And so in that, you can start asking what bacteria are there, but you don't really know spatially how they are organized. And so that is why 3D-omics came into fruition, where we had the aim of developing tools that we could study the microbiome of animals in a very spatially aware manner. And so we're processing samples such that the spatial data is still taken into account, so that we can now start asking questions of who exactly is where. So this is the general workflow of our whole project, and it's a very top-down approach because it starts all the way from animal trials, where we subject different animals to pathogenic challenges, as well as probiotic treatments, and also different types of feeding trials. And so then what happens through the trials is we start sacrificing animals and sampling their gut segments. And so my role in the project is actually in histology, where we slice these gut samples kind of like a loaf of bread. And then these become cryosections that we can then further laser microdissect with a laser microscope to take very, very, very small samples in a very spatially manner. And so then we apply a multi-omic approach, where we look at the DNA contents of these very small samples, as well as the metabolites and eventually RNA. And then also in the project, there are people who do modeling based off of the data that we have to figure out whether or not these spatial microbiomes can predict the outcomes of future animal data. And so such a project, top down, all the way from animal trials to modeling, definitely couldn't be done by a single lab. And so this project actually brings together partners from all over the EU and the world actually. And so it's really bringing experts in their various fields and kind of creating this ambitious project to make a pipeline to study livestock microbiome. And so it's also good to note that we have industrial partners that provide us with the probiotics because they would be interested in knowing how their probiotics actually impact the microbiome landscape. So again, this is a zoom in on the sampling part of the workflow. And so again, we go from animal to gut segments to cryosections to microsections. And my role is actually in the project a microscopist. And so there are two forms of microscopy that we're using. One is confocal fluorescence microscopy, and the other actually is a laser microdissection microscope that was made specifically for this project, and it's one of its kind, which is pretty cool. And so just a reminder on scale, we're studying bacteria that are in a chicken, and we're doing this by cutting very, very small samples of the chicken that's equivalent to 1/4,000,000th the size of a huge chicken. And so if we're studying such small things, we need to have the right tools. And so I grew up in the era where a lot of people played with glow-in-the-dark toys. And recent surveys, like when I've talked to other students and different classrooms, it's still good that the children are still playing with glow-in-the-dark toys. And I have a core memory of myself because I am/was afraid of the dark. And so I really needed to have glow-in-the-dark stars on my walls in order for me to fall asleep. But when I transitioned into science and microscopy and imaging, I was actually pretty excited that this type of technology in the toys is being used also in microscopy. And so here are just some images of fluorescence microscopy where our targets are making fluorophores, fluorescent proteins that we can now localize and ask questions about where are these bacteria being made, what is being made, and other things like that. Yeah. And so I'll briefly talk about the method that I was developing in the 3D-omics consortium. We were hoping to create a multiplexed FISH pipeline. And so FISH stands for fluorescence in situ hybridization. It's not the animal, which is a bit confusing because we talk about chicken, swine, and then fish. But briefly, fluorescence in situ hybridization is basically the employment of fluorescent DNA probes to fish out different DNA or RNA targets. And so this is usually used in vivo samples because in vitro you could make a bacterium, produce these fluorescent proteins and just look at where they are. But when you're looking at animal samples where you don't have the genetic pliability, fish is a great way to do this. And so here I'm showing some examples. Fish is often used for karyotyping. And so in order to figure out the number of chromosomes that an individual has, they use these fluorescent probes to figure out what genetic profiles are in each chromosome. And then also in the middle, we've all seen the development of a Drosophila embryo. And this is the fluorescence version of it, where you see the notch and the Hox genes that are patterned throughout. And so lastly, on the right is a fish image of a diseased organ where you see there's bacteria that has now penetrated the liver. Yeah. And so fish is really neat because, like I said, you're looking for targets that are in your sample, and you can start asking questions of who all is there, what bacteria is there, how many of them are in a fixed area, and also who is next to each other. And so actually my Zoom background is an example of a fish image, and we'll get into the analysis of that later. And so briefly, fluorescence in situ hybridization involves the permeabilization of bacteria. And so the first step is to open up your bacteria, because if you just threw DNA at bacteria, they can't go into your targets. So this involves getting rid of proteins on the surface of the bacteria. Then we employ our fluorescent probes, and they will go towards your targets, but oftentimes there'll be off-target effects. And so then we have a washing step to get rid of the promiscuous probes. And then imaging will now then light up the bacteria that you are targeting. And so here, for example, we are targeting the rod-shaped bacteria. They give you a green fluorescent signal, while the cocci bacteria do not give you this signal. And so in one of the first trials that we did in 3D-omics, we asked the question of how Salmonella infantis infection changes the landscape, and also how the probiotics from the industrial partners would change the landscape. And so what I did was I designed a panel of 10 different fluorescent probes, multiplexed with different fluorophores, such that we can look at how these gut samples look and how these bacteria are organizing within the gut. And so here I hope you can appreciate from the left is a healthy adult chicken. And there is the most healthy adult chicken cecum. So that is our target organ. And the reason is because we knew that this organ had the most diversity of bacteria. And so you can see there's all sorts of different colors, and there's pretty large diversity in the healthy chicken. But then once there's a Salmonella infection, you actually see now there are aggregates that are all throughout this chicken segment. And these aggregates are actually of Blautia bacterium. And we were excited to see this because it meant that just a singular bacterium pathogen shifts the landscape very dramatically. And so now diversity has dropped, but also the organization has changed as well. And then if you move to the right, I hope you can see that now with the probiotic, the Salmonella infantis bacterial load actually drops, and the diversity also drops, which means that the probiotic is shifting the landscape to favor 2 bacterial clades to combat and get rid of the Salmonella infantis. And so if we zoom in, you can also see in the healthy chicken, you see a lot of colorful bacteria, but also there are aggregates, but not to the size of those that are in Salmonella. And so here on the left, you're seeing an Eisenberghalia aggregate that's pretty small, whereas in the Blautia aggregates in the Salmonella infection, they're way larger. So something about Salmonella infantis infection shifts it such that it's preferring to allow Blautia to aggregate and grow. And then lastly, with the probiotics, you see here these chain-linked bacterium. These are also Blautia, but a different species. And so somehow the shifting away from the Blautia to a different type of Blautia actually protects the chicken from survival. Yeah, so these are kind of qualitative descriptions of what we see using fluorescence in situ hybridization on the different chicken. But another thing that we had to do was to basically count the bacteria. And so one of the first steps is taking an image of these fish images, doing segmentation to figure out what is a cell, what is not a cell, what is noise, etc. And then the last step is classifying based off of the fluorophore intensities, their identity of each bacterium. And so I like to show this slide also to say that like a lot of the AI image recognition things that we do nowadays, like Google image reverse search, it all depends on segmentation, because the AI and the computers are able to tell a horse is a horse based off of the outline and segmentation of a horse. And so applying these type of things into our image analysis also helps. And so then we are able to start asking questions of how many of each genera of bacteria are there. But we can also start now using different measurements to ask the question of, if this bacterium is here, how often is this other bacterium being shown? And so this way we are able to get a very spatially single cell level of questioning and quantifying of the microbiome in different contexts. Yeah. And so now I will pass on the time to Amalia, who will talk to you all about the more DNA side of our method.
Thank you, Bryan. And you can hear me? Yes, good, great. Okay, so I will also start with a, okay, let me fix. I will also start with a small introduction to what is the microbiome. And as we have said many times today, it is the community of microorganisms such as bacteria, archaea, fungi, parasites, viruses that one can find in any and specific environment, such as the animal gut. And depending on the environment, depending on the sampling site, there can be hundreds of different microbes, present. And this is an example. This is looking inside the chicken gut. And we know that in here, there are hundreds of different bacteria, but just by looking at the microscope, we cannot see them and characterize them and describe them. So how can we describe a community that is like so small, yet so complex? And traditional microbiology methods really fail us when it comes to complex microbiomes, because you can try to look at the bacteria at the microscope, but if you have too many that are different but look very similar, you will not be able to differentiate them. Similarly, you can try to grow the bacteria and look at the morphology of the colonies, but again, if you have a complex community with multiple different species, this is not possible. Likewise, you can try to differentiate between the bacteria by growing them in differential medium or selective media and doing different biochemical tests. But still, this is not really working for the microbiome because not all the microbes are culturable. And also, even if they were, it's extremely time consuming to isolate each and every one of them, grow them, and test them. So we need a better way to do this. And this is the DNA, because the DNA can tell us exactly who is there. It is a signature of each and every organism in our microsample, in our sample. One way to do this molecular identification is by amplicon sequencing, sequencing the 16S gene. This is very convenient as it can really tell us who is there, and you don't need any culturing, it's very fast. However, you cannot identify very well down to species level resolution and not at all below species level. And also because you only sequence one gene, you cannot detect any functions such as antimicrobial resistance or polysaccharide degradation. Another way to do the molecular identification is by sequencing the genome, the whole genome of the organisms inside our sample. And when you sequence the genomes of multiple organisms at once, this is called metagenomics. And this is great because unlike traditional microbiology, you don't need to culture anything, it's very fast and high throughput. And unlike 16S sequencing, you can now go down to strain level resolution. And you can also have functional annotations of your genomes and know if you have genes that do specific things in there, like antimicrobial resistance genes. So this is what we use at 3D-omics. And this method, we mostly use it at the University of Copenhagen, but we have other partners in the consortium that do metatranscriptomic analysis and metabolomics analysis, which means that they put other layers of information in the same samples that we analyze as well. So I will also talk about microbiome scale, like Bryan did a bit. So usually when you study a microbiome sample, you have quite a big sample. So if we want to study the cecum of a chicken, we would get the tissue and then get with a loop quite a bit of the material inside the cecum, and then save it in a tube and then extract the DNA. And you would usually get a lot of DNA from a sample like this, which then allows you to sequence it in many ways. Usually we do short read sequencing, but we are also doing some long read sequencing, as they both have pros and cons. And then from those sequences, we can assemble the genomes of each of the organisms inside the sample. And then we can make a catalog of all the genomes that we found, which is basically a database of all the microbes, of all the organisms that exist in our chicken. This catalog we can use for a lot of things. One of the uses is for designing the probes for fish that Bryan showed you before in order to visualize the 10 most abundant microbes in there. But also we use the catalog in order to quantify the number of genomes for each bacterium that we find in the conventional samples. And this way we get some general insights about how the microbiome is affected by the different treatments that the animals have gone through. And this is already a lot of data, and we can work with it very well, but we went one step further in 3D-omics. And instead of just using the conventional samples, we now started also using microsamples. So for this, we start again with the same animals and the same tissues, a different segment of the same tissue. And for example, the cecum, then we slice it, we put it on a microscope slide, and then using this laser microdissection microscope that Bryan showed you before, we can cut tiny pieces that are these little circles. And these microsamples, we collect each of them inside little lids. And it looks like this in the microscope. They are tiny. And we always check whether we have collected the microsample. And they are, of course, invisible to the naked eye. And we have estimated that each microsample only contains around 1,000 cells, bacterial cells, which means there is very, very little DNA in there. And this causes us a lot of challenges, and we have spent a lot of time optimizing the protocols to work with such small amounts of DNA, such as we found out that we should only do lysis of the cells to release the DNA instead of doing an actual DNA extraction. We try to move our microsamples as little as possible so that we don't lose any material. And we have adapted all our reactions and protocols to work with this very small amount of DNA. And of course, we have included many, many, many negative controls to make sure that any signal we get is real. So our partners in 3D-omics, they are veterinarians for the most part, and they have run many experiments on swine to see how fiber and protein in feed affect the animals, and then we check what happens in the microbiome. And our other partners have checked what happens in poultry health, for example, checking whether a symbiotic supplement can help against salmonella infection, or whether vaccination against the histomonas can help the animal survive the infection better. And overall, we have received several samples from more than 460 animals from our 3D-omics partners. And this causes a small problem when we scale up for the microsamples, because when we are doing the conventional metagenomics, one sample from one organ, we need one sample for one organ for one animal, whereas when we do the microsamples, we need at least 80 microsamples per organ per animal. So this scales up extremely fast. And if we look at the timeline of our work, we spent first some time developing the method to be able to do metagenomics in such small microsamples. And then we got a robot which actually helped us process thousands of microsamples quite fast. And up to now, we have processed more than 4,000 microsamples. And with this robot, we usually process 182 microsamples per day, which is as many microsamples we can cut with a laser in a day. And now, why do we do this? Why do we go to the microscale? What does it give us extra than the conventional samples? So the first thing is that we have found that with the current methods that we're using, in the microsamples, we find more bacterial species and bacterial genera that we didn't capture in the conventional samples. And this is because they're very rare and they get the chance to be sequenced when the microsample is very, very small. What is the most important addition of for the micro samples is that it gives us spatial context. So for example, if we would have two different treatments here and we would look at their microbiome, now we know by this photo that in the second treatment there are some aggregates, that these bacteria go closer together and these go closer together in the second treatment, which doesn't happen in the first treatment, right? But then if we sample with the conventional method, all of these bacteria and all of these bacteria will be mixed up with each other, and the final result will look the same for both animals, even though the treatment did change the structure of the microbiome here. Whereas if we use the microsample method, then the microsamples can capture the different composition and the aggregates that are now found in the treated group. And we do see this in our real samples. And we have started combing through the data and see which relationships are actually changing when we look into the microsamples to investigate them further. Another thing that we can do with the microsamples is that because we keep a note of every coordinate of every microsample that we cut, then we know the physical distance between microsamples. And because we have sequenced them with this whole genome sequencing, we also know who is in each microsample and what they do. This means that we can plug all this into an analysis model, and then we can see if there is any spatial structure in this tissue. For example, in some tissues, there is no spatial structure. All the microbes are mixed up together. In this particular section of the colon, then we found some spatial structure. So the next steps for this part of the project is that before the summer holidays, we will have finished the production of around 6,000 microsamples. And then we can start analyzing everything and supplementing the analysis of the conventional microbiome samples with our insights from the microsamples. So thank you for me, and thank you for listening, and of course thank you to the entire 3D-omics consortium for the samples and the collaborations. Thank you.
Thank you both. Super interesting presentation. I know we've got one question in the chat, and then I've also had a couple of questions sent to me directly. So maybe Aurel, do you want to say your question out loud, or should I read it? If you want to say it, unmute yourself. Okay, yeah.
Yes, I can say it on live. I would like to know the main core difference between metagenomics and metatranscriptomics.
Yes, in the metagenomics, we sequence the DNA, whereas in the transcriptomics, we sequence the RNA. Often we turn it into DNA in order to sequence it, but the initial target of the nucleic acid is the DNA in the genomics and the RNA in the transcriptomics. So in the genomics, we can see what is the potential of the genes that are there to potentially be used, for example, an antimicrobial resistance gene. In the transcriptomics, we could see if this gene is getting expressed. So if it is really being used by the bacterium, or if whereas in the genomics, we just see the potential of it getting used. Does it make sense?
Yeah, makes sense. So as I understand, genomics just give the potential, while transcriptomics give the really express gene, right?
Mm-hmm.
Okay, good. Good to know.
Okay, thank you both. Then I have a question that was sent to me directly. So how do you see this multi-omic approach potentially impacting livestock health management in the future?
Maybe I can start and then Bryan can answer as well. Okay, what I was thinking while hearing, for example, David's presentation before is that sometimes you expect to see a difference in the microbiome and then you sample it and then you don't see a difference. And I think that by using all these different layers, we can see eventually if there is truly a difference. So, for example, if we just do genomics, but what changed by the treatment is just the expression of the genes, then we will capture it with the transcriptomics. If what we see is a different structure in the spatial structure in the bacteria, but the same bacteria are there, then just doing conventional metagenomics or conventional anything will not show any difference. You have to go into the microscale level to see the spatial differences. So ideally, we want to see all the layers to understand exactly what happens, even if it looks like there was no difference maybe at the beginning. Maybe Bryan wants to.
I think also the part of 3D-omics being spatial is very important because I believe a lot of times when and this is known in the field, that like symbiotics, probiotics, the establishment of these cultures are not necessarily always that good, but we know kind of through trial and error that these work. And so the hope from 3D-omics is to maybe re-strategize the way we use probiotics and symbiotics on whether or not there are other methods to try and anchor these probiotics to the villi, for example, or whether or not these probiotics are better when they're in the lumen. And so these are things that we can do in improving our treatments in like whether or not looking at a spatial manner, because up until now, most of the times we are just guessing that putting a probiotic or symbiotic would allow for improvement, but we can also apply a more spatial method into improving that. So refining our strategies, kind of.
Okay, thank you. That's very interesting and cool as well. And then I'm just going to send the 2 extra questions I got as well in the chat and read them to you both as well. So the first one was, can the spatial microbiome mapping techniques developed in the 3D-omics project be applied to rumen microbiome studies to better understand microbial colonization? Does that make sense? Yeah.
I think that's also linked to what Endris had said about how this type of technology can be applied to even the nematodes and protozoa. And so the idea actually is our methodology is not limited to what we have been asking. And actually, the methods truly is using a spatial low input nucleotide methodology. And so both of the questions relating to protozoa as well as the rumen, we would love to do as well, because applying a spatial aspect to a lot of the already studied organs can actually reveal a lot more other things. And Amalia already showed that we are now detecting things that conventional methods don't even pick up on. Yeah.
And to add on that, in the case of the question about the protozoans and the nematodes, already in the samples that we have collected and sequenced the DNA, we have sequenced the DNA that was present in the sample of all the organisms that were there. Currently, we are focusing on analyzing the bacterial DNA, but we have all the data. So if we would like, we can go back and look for nematodes and look for protozoa or viruses. But it's not right now the target of the project, but the data is there. So if we want to analyze it, we can anytime go back to it, which is a great thing about using metagenomics, because you amplify, you sequence everything in there. And regarding on whether we can run this on rumen, we haven't tried this in 3D-omics. As Bryan said, we can totally apply it to rumen, and it would be, we believe, beneficial to look into that. Sometimes it is challenging when the animal is very big to actually get good slices for the microscope, but I guess there are workarounds for that. Yeah.
Okay, interesting. Thank you. And then the last question I have here is, can the multi-omic approaches discussed help develop more targeted interventions for early life animal microbiome development?
Yeah.
I guess that's eventually the goal we want to get at.
Okay.
But I think what the project has revealed, and I think this has been shown in humans and almost all animals, where like early life complexity in bacterial microbiomes are always less, and then eventually in adulthood, they become more diverse. And so whether or not maintaining diversity is always good is still a question that we have in the project, because we've revealed that probiotics actually shift diversity a little bit less, but somehow it still protects the animals. And so it is an eventual goal of the project, but we probably are not directly there yet. Yeah.
Yeah.
Okay.
We have, our veterinary partners have tried to actually sample through the early life of the animals. I also think we're not there yet, but we have tried like for example, the swine experiments, they did start changing the feed when the animal was very young in order to see what happened when it grew up. The vaccination against histomonas happened when the animal was very young, and then we infect later in life. Same, the supplementation with probiotics happened since the animal started feeding, so super young, and then the infection with salmonella happened a bit later. So there is always a component of changing the microbiome early and then seeing what happens later. And we usually have samples across the lifespan of the project. However, because me and Bryan, what we study is inside the gut, so we need to dissect the actual animal. Somebody needs to kill the animal for it. Then we cannot study the same animal across time. We study the same group of animals from very early until they are dissected. I hope that makes sense as an answer as well.
Yeah, it makes sense to me at least, and I'm from a social science background, so hopefully other people can understand as well. Yeah, if there's any more questions, feel free to unmute, turn your cameras on, pop in the chat. In the meanwhile, thank you both again for a very interesting presentation. Thank you for all the participants today for coming. Very good discussions and some great questions. Yeah, I'll give it 1 more minute for any more questions that might come in. Okay, looks like we're out of luck. And then just again, this is the last webinar before the summer break. If you want to be notified for the next one, do feel free to register for the HoloRuminant newsletter. Will you be notified? You can also follow their project on socials where we promote them as well. And you'll also get the link for this in the chat. And to discover more of the other projects, you can scan this QR code or also follow the link that will be in the chat as well. You can also find all of these projects on social media. If you go to HoloRuminant, the latest posts all have the other projects tagged as well if you have any trouble finding them. Yes, so thank you all for joining today and for all our speakers and for all the great questions we had. And let us know if you have any questions later on over email. And this will also be recorded as it's been today and put on the HoloRuminant YouTube channel. And there you can also see all the previous episodes as well.